This is a presentation about Alzheimer's disease given by Robert Cowles, James Burrell, and Kyle Hamilton. Okay, so basically, Alzheimer's disease is when degeneration of the brain causes memory loss. Usually occurs in older people over 65 years and older. And the thing is, scientists don't really know how to prevent this or exactly how it occurs.
Many believe that Alzheimer's disease is passed through genetics. And in this first study we looked at was a twin study, and what they examined was heritability. Heritability is how much variation in an obvious and observable trait in a population is due to genetic variation among individuals in that population. And so the study, they used monozygotic twins, and they share the same genes, and dizygotic twins, which share on average 50% of their segregating genes. And so what they looked at was, so they screened the individuals for Alzheimer's disease, and if one of them was suspected to have it, their co-twin received a complete clinical diagnosis evaluation for Alzheimer's disease. And then from there they estimated heritability from that data. And so what they found was heritability for Alzheimer's disease was estimated to be 58% in the full model and 79% in the best fitting model. And so what that means is that heritability for Alzheimer's disease was high, right? And the next study was a genome study. And so what they looked at was genes. So using a two-stage genome-wide association study, they're able to replicate and establish association with an APOE gene, which is expected to be a significant factor to cause Alzheimer's. And they also observed two genes that were not previously associated, the APOJ and PCALM SNPs. And then they repeated the experiment, and everything, the associations from stage 1 were replicated. And so this diagram is a diagram of the genes from the genome-wide study. And so what we can see here is that they have a p-value listed on this y-axis and the genes on the chromosomes on the x-axis. And as we can see, this APOE gene is well above the red line, which is a p-value of 9.4 times 10 to the 8, which is actually the threshold for genome-wide acceptance, and as well as this PCALM and CLU SNP. So we can see that there is some significance, and pretty obvious to see these genes stick out from this p-value.
Beta amyloid is an insoluble, sticky peptide that is constructed from 36 to 43 amino acids. It is a derivative of APP, which is amyloid precursor protein. Little is known about the function of APP, but it is thought to help attach cells to one another. It also has a possible function of arranging neurons in the early development of the brain, which is the origin of amyloid beta. Amyloid precursor proteins split into amyloid beta and SAPP, soluble amyloid precursor protein. The reason beta amyloid is the main target for research is since it is still insoluble, unlike SAPP. A mutation in the APP replaces amino acid valine with isoleucine either leads to increased production of amyloid beta or production of longer, stickier amyloid beta. Either the increased production mutation or the stickier peptide mutation then leads to plaques at the site of synapses by building up excess amyloid beta. These plaques, known as brain plaques, impede movement of axonal signals, which lead to deterioration of brain activity.
As well as just looking at the protein and the correlation between it and Alzheimer's, scientists also looked at beta amyloid clearance. So in this study there were 12 patients who had Alzheimer's disease, and the other 12 participants were not diagnosed with it. And so using a metabolic labeling process, they were able to measure production and clearance of beta amyloid 40 and 42. And so a person can clear beta amyloid. They can clear it by enzymatic pathways. So it would be, you'd have specific beta amyloid degrading enzymes or non-enzymatic pathways. So example of that would be you would transport it across blood vessel walls into circulation. So after data collection, they found a non-significant difference between beta amyloid production between Alzheimer's patients and the controls. However, they found a significant difference between the clearance of beta amyloid, 40 and 42. So for Alzheimer's disease patients, they could clear beta amyloid 40 at 5.2% per hour, as opposed to the controls who could clear it at 7% an hour. In the case of beta amyloid 42, Alzheimer's disease patients could clear it at 5.3% per hour, and the controls were able to clear it at 7.6% per hour.
Tangles refers to bunching and crossing of transport systems. Another protein associated with the occurrence of Alzheimer's is the tau protein. These proteins just function to keep the pathway straight. When the proteins no longer function correctly, the pathways become inefficient and lead to decreased signaling in the brain through neural pathways. The motor protein kinesin, which is involved in moving the signals through the pathway, become obstructed and cannot carry out the functions they previously served. Kinesin is also the protein believed to be responsible for the movement of microtubules in the process of mitosis and meiosis. The process of moving these signals in the axon is known as cargo transport. The figure here displays what the axonal swelling due to amyloid beta brain plaques look like. A normal axon is the A line on the far left, which is functioning fine with no plaques present. As you move right, you begin to see an increase in size and number of plaques along the pathway. These plaques are also considered axonal swelling. These pictures just show a different view of the same type of phenomenon. The axonal swellings are either circled in red or pointed out by the red arrows. The experiment for which these pictures were sourced studied the activity of the brain and plaques in mice and humans. The kinesin 1 motor proteins were decreased to observe the effect they had on the axonal swellings. The lab mice, which are the TG-SWAPP-PRP, have brains more affected by Alzheimer's than the wild types. The wild types were just used as a control to demonstrate the function of a normal brain and pathways. Figure C shows the increase in swellings in different regions of the brain, and the data suits the experiment and theory well. The entorhinal cortex is the cortex responsible for memory, showing a correlation between axonal swellings and memory disabilities. Figure C just shows the difference in amyloid beta ratios for the reduced kinesin brains and the regular kinesin brains. This again shows the effect kinesin reduction has on the brain, and this time shows specifically which regions and which beta is affected the most.
There are no known cures for the disease, but there are some treatments, some for memory loss, which would include cholinesterase inhibitors, which prevent the breakdown of acetylcholine, which is an important chemical messenger involved in learning and memory, and memantine, which regulates the activity of glutamate, which is also another chemical involved in learning memory. Yeah. And for behavioral changes, they treat depression, anxiety, hallucinations, etc. They'll just give you like antidepressants, anti-anxiety medication. And to treat sleep changes, there's both non-drug and drug approaches to this. So with the drugs, you could take like sleep medication or anti-anxiety medication could help. And in non-drug treatment, they try to change sleeping patterns. And finally, there are alternative treatments like herbal remedies, dietary supplements, medical foods that they just claim will help this somehow. Anyways, they're basically based off other people's words or testimonials, and there's very little scientific research about this, so it's just kind of going around by word of mouth. So just as a take-home message, we want to say that Alzheimer's disease is rampant throughout the world, and it is most likely caused by these beta amyloids blocking cell signaling between the synapses in your brain. Although there are no known cures, there is extensive research being done to further understand the role of beta amyloid and why some people have a more difficult time clearing it than others, and how we can prevent this disease altogether.